Every single person watching this video right now carries a molecular passport inside their cells, a genetic document that records the exact route your ancestors took across continents over the past 200,000 years. But here's what makes this passport extraordinary: it only comes from your mother. And her mother. And her mother before that, in an unbroken chain stretching back to a single woman who lived in Africa roughly 150,000 years ago. Scientists call her mitochondrial Eve. But if we all descend from one woman, what happened to everyone else who was alive back then? Why does this genetic trail contradict what we thought we knew about human migration? And most unsettling of all, why do some indigenous populations, who've lived on their land for 50,000 years, show DNA markers that suggest they came from somewhere completely different? The answer rewrites human history. And it's hidden inside the powerhouse of your cells. To understand how we discovered humanity's greatest journey, we need to go back to 1963, when a discovery in a Swedish laboratory would eventually unlock secrets that had been buried for millennia. That year, Margit Nass and Sylvan Nass, two researchers studying cell biology, made an unexpected observation: mitochondria, those tiny energy factories inside every cell, contained their own DNA. This was revolutionary. For decades, scientists believed all genetic information resided in the cell's nucleus. But here was a completely separate genetic system, operating by different rules. The truly fascinating part? Mitochondrial DNA, or mtDNA, is inherited exclusively through the maternal line. When a sperm fertilizes an egg, the father's mitochondria are destroyed. Only the mother survive. This means your mtDNA is identical to your mother's, which is identical to her mother's, going back through an unbroken female lineage. But it took another 2 decades before anyone realized what this meant. In 1987, a team led by Alan Wilson, Rebecca Kahn, and Mark Stoneking at the University of California, Berkeley published a paper that would shake the foundations of anthropology. They analyzed mtDNA from 147 people representing diverse populations across 5 geographic regions. The study was titled Mitochondrial DNA and Human Evolution, but the media had a catchier name for their discovery: they'd found mitochondrial Eve. The researchers noticed something extraordinary in the patterns. All modern human mtDNA could be traced back through a branching tree, a phylogenetic tree that eventually converged on a single point: one woman who lived in Africa approximately 200,000 years ago. But here's where it gets confusing, and where most people misunderstand what mitochondrial Eve actually means. She wasn't the only woman alive. She wasn't even necessarily special. There were thousands, possibly tens of thousands of other women living at the same time. But through the random process of genetic drift, where some lineages produce only sons, or no children at all, every other maternal line eventually died out. Mitochondrial Eve is simply the most recent common matrilineal ancestor of all living humans. Think of it like surnames. In any given town, you might have dozens of family names. But over centuries, some families have only daughters, some have no children, and gradually, certain surnames disappear. Eventually, if you went back far enough, you might find one surname that all current families connect to, not because that family was better, but because through random chance, theirs was the line that survived. But why was this discovery so controversial? Because it directly contradicted the dominant theory of human evolution at the time. For most of the 20th century, anthropologists were divided into 2 camps, each with radically different views of human origins. The first camp supported the multi-regional hypothesis. This theory proposed that modern humans evolved simultaneously in different parts of the world: Africa, Europe, and Asia, from earlier hominid populations that had already spread across the globe over 1 million years ago. According to this view, Homo erectus left Africa, settled in various regions, and then slowly evolved into modern Homo sapiens through parallel evolution, with enough gene flow between populations to keep them as one species. The competing theory was called recent African origin, or what we now call the out of Africa hypothesis. This model argued that modern humans evolved recently in Africa somewhere between 200,000 and 100,000 years ago, and then migrated outward, completely replacing all other hominid populations they encountered. Neanderthals in Europe? Replaced. Homo erectus in Asia? Replaced. This wasn't gradual evolution across continents, it was a single population explosion from one source. The stakes were enormous. These weren't just academic theories, they shaped how we understood human diversity, race, and our fundamental relationship to each other. The multi-regional hypothesis suggested deep genetic differences between populations that had been separated for over 1 million years. The out-of-Africa model suggested we were all far more closely related than we thought. The 1987 mtDNA study didn't just favor out of Africa, it provided a molecular clock, a way to date when these migrations occurred. Here's how that worked. mtDNA mutates at a relatively predictable rate. By counting the number of mutations between different populations and calibrating against known evolutionary events, researchers could estimate when those populations diverged from each other. The genetic diversity was highest in African populations, suggesting they'd had the longest time to accumulate mutations. Non-African populations showed less diversity, indicating they were younger, having descended from a small group that left Africa more recently. The dates lined up with an exodus from Africa roughly 70,000 to 50,000 years ago. A small group, perhaps just a few thousand people, crossed from the Horn of Africa into the Arabian Peninsula. This founding population carried only a subset of Africa's genetic diversity. Everyone outside Africa today descends from them. But the story wasn't complete. The question became, where did they go next? And how could we trace their roots? As mtDNA research exploded in the 1990s and early 2000s, scientists began identifying distinct lineages called haplogroups, branches on the human family tree, each marked by specific mutations. These haplogroups became breadcrumbs, allowing researchers to reconstruct the exact paths humans took as they populated every corner of the planet. The story that emerged was staggering in its scope. The journey began in East Africa, where the oldest and most diverse mtDNA haplogroups labeled LaRho, Lone, LT-double, and L3 still exist today in populations across sub-Saharan Africa. These lineages date back over 150,000 years. For tens of thousands of years, early humans remained in Africa, developing language, tools, and culture. Then, sometime around 70,000 years ago, something changed. A small group carrying haplogroup L3 left Africa, crossing into the Arabian Peninsula. This timing is critical because it coincides with a period when sea levels were lower, creating land bridges and coastal routes that would later be submerged. Archaeological evidence suggests they followed coastlines, living off marine resources, a strategy that would allow rapid movement along predictable food sources. From the Arabian Peninsula, the trail splits. One branch moved east along the Indian Ocean coast, reaching Southeast Asia remarkably quickly, possibly within just a few thousand years. These migrants carried haplogroup M, which would go on to spawn dozens of subgroups throughout Asia, Oceania, and the Americas. The speed of this coastal migration was breathtaking. By 65,000 years ago, humans had reached Australia, crossing open water that required sophisticated boats and navigation skills. The Aboriginal Australians represent one of the oldest continuous cultures outside Africa, and their mtDNA shows they descended from those first coastal migrants. Meanwhile, another branch moved north into Central Asia and Europe, carrying haplogroup N. This lineage would diversify into the haplogroups now common in European, Middle Eastern, and some Asian populations. The European settlement happened later, around 45,000 years ago, when humans carrying haplogroups U and H moved into a continent already occupied by Neanderthals. And this is where the story gets complicated. The mtDNA evidence showed modern humans replacing other hominids, but it was too clean. If modern humans and Neanderthals lived side by side in Europe for thousands of years, wouldn't there be some interbreeding? The mtDNA data suggested no, there were no Neanderthal lineages in modern mtDNA. This supported the complete replacement model. But that conclusion was about to be challenged in a way no one expected. The final leg of humanity's journey led to the Americas. Analysis of Native American mtDNA revealed something striking. All indigenous American lineages belong to just 5 haplogroups A, B, C, D, and X. These groups trace back to populations in Siberia and East Asia. The data suggested multiple waves of migration across the Bering Land Bridge, a massive landmass called Beringia that connected Asia to Alaska during the last ice age. The first wave crossed around 15,000 to 20,000 years ago, rapidly spreading south along an ice-free corridor and coastal routes. Within just a few thousand years, humans had reached the southern tip of South America, a distance of over 10,000 miles from the entry point in Alaska. The genetic evidence showed founder effects, sharp reductions in genetic diversity indicating small founding populations that exploded demographically once they entered unoccupied territory. By 12,000 years ago, humans had reached every inhabitable continent. mtDNA had mapped the journey in unprecedented detail. The evidence seemed overwhelming. We were one species, with one origin, and one incredible story of exploration. But science is never finished. And in the early 2000s, a new technology was about to expose the limits of what mtDNA could tell us. In 2010, a team led by Svante Pääbo at the Max Planck Institute announced something that should have been impossible. They had sequenced the genome of a Neanderthal, not just the mtDNA, but the entire nuclear genome from a 40,000-year-old bone found in a Croatian cave. The results were shocking. Comparing the Neanderthal genome to modern humans revealed that non-African populations carry 1% Neanderthal DNA in their nuclear genomes. Europeans and Asians had Neanderthal ancestry. Africans did not. But here's the mystery. Remember, mtDNA showed no evidence of Neanderthal maternal lineages. How could we have Neanderthal ancestry without Neanderthal mtDNA? The answer reveals the fundamental limitation of mitochondrial DNA. mtDNA only tracks one line, the maternal line. It's like trying to understand a tree by following a single branch from leaf to root, ignoring every other branch. Nuclear DNA, by contrast, is inherited from both parents, capturing the full complexity of ancestry. What likely happened is this: when modern humans and Neanderthals interbred, and the evidence now shows they definitely did, most of the hybrid offspring didn't survive, or didn't reproduce successfully. This is common when closely related but distinct species interbreed. However, some nuclear DNA fragments provided advantages and were preserved by natural selection, while the Neanderthal mtDNA lineages died out through genetic drift. Think about what this means. The mtDNA map of human migration wasn't wrong, but it was incomplete. It showed the highways of human expansion, the successful maternal lineages that survived. But it missed the side roads, the dead ends, the populations that contributed genetically but whose direct maternal lines vanished. More discoveries followed. In 2010, another ancient genome was sequenced from a finger bone found in Denisova Cave in Siberia. This represented an entirely unknown hominin group now called Denisovans. And modern humans carry their DNA too. Melanesians, Aboriginal Australians, and some Southeast Asian populations have up to 5% Denisovan ancestry. Again, no Denisovan mtDNA exists in modern populations, but their nuclear DNA persists. The picture became more complex still. In 2015, researchers discovered that West Africans carry DNA from an unknown archaic hominin, something that interbred with modern humans in Africa before the Great Migration. Once again, mtDNA had missed it entirely. This wasn't a failure of mtDNA analysis. It was a reminder that evolution is messy. Populations don't evolve in isolation, they meet, mix, and sometimes merge. mtDNA provides one powerful line of evidence, but the full story requires multiple sources: nuclear DNA, Y chromosome analysis, archaeological evidence, and more. Yet despite these limitations, mtDNA remains uniquely valuable for one simple reason: it's abundant, it mutates at a predictable rate, and it tracks direct maternal ancestry with no recombination, meaning the signal doesn't get scrambled with each generation the way nuclear DNA does. But the breaking point wasn't just scientific. It was cultural. As mtDNA testing became commercially available in the early 2000s, millions of people began exploring their genetic ancestry. Companies promised to reveal your ethnic makeup, trace your maternal and paternal lines, and connect you to ancient populations. It was marketed as a journey of self-discovery. But the results often challenged people's understanding of their own identity. Indigenous communities found genetic evidence suggesting their ancestors arrived from distant continents, results that sometimes contradicted origin stories passed down for thousands of years. White Americans discovered African ancestry. People who identified as fully European found Native American or Asian haplogroups. The molecular passport didn't always match the cultural passport. Some indigenous groups rejected genetic findings entirely, arguing that Western science was imposing its framework onto communities with their own valid historical narratives. Who gets to define where someone comes from a DNA test, or the stories and traditions maintained by communities themselves? The tension revealed something profound: genetic ancestry and cultural identity are not the same thing. Your mtDNA might say your maternal ancestors walked out of Africa 70,000 years ago, moved through Asia, crossed Beringia, and settled in the Amazon 12,000 years ago. But what does that mean for your identity today? Does it matter if your community has lived in the same place for 500 generations? There's also the problem of interpretation. Commercial DNA tests often assign ethnicity percentages based on comparing your DNA to reference populations. But these reference groups are modern constructs. Saying you're 30% Italian doesn't mean your ancestors lived in modern Italy. It means you share genetic markers with people who live in Italy today. Ancient populations didn't recognize these borders. They were constantly moving, mixing, and migrating. Even more troubling, the mtDNA data has been misused to argue for racial hierarchies. Some have pointed to genetic clustering of populations and claimed it justifies racial categories. But genetic diversity is clinal. It changes gradually across geography. The differences between populations are far smaller than the variation within populations. Two individuals from the same ethnic group are often more genetically different from each other than either is from someone of a completely different ethnicity. The fallout from mtDNA research taught us that genetics is descriptive, not prescriptive. It can tell us where people moved and when they mixed, but it can't tell us what those movements mean or how we should identify ourselves today. Science has attempted to address the limitations of mtDNA by integrating multiple genetic systems. Y chromosome analysis tracks paternal lineages. Autosomal DNA, the chromosomes inherited from both parents, provides a fuller picture of ancestry. Ancient DNA from archaeological sites fills in gaps that can't be seen in modern populations. But mysteries remain. One of the most persistent, the peopling of the Pacific. Polynesians accomplished one of humanity's most impressive feats, sailing across thousands of miles of open ocean to settle remote islands like Hawaii, Easter Island, and New Zealand. mtDNA shows they descended from populations in Taiwan and Southeast Asia, part of the Austronesian expansion that began around 5,000 years ago. But how did they navigate? How did they know islands existed beyond the horizon? And why do some Polynesian populations show genetic markers linking them to South America, suggesting contact long before European exploration? In 2020, a study found that some Polynesians carry Native American DNA dating to around 1200 CE, centuries before Columbus. This suggests either Polynesians reached South America or South Americans reached Polynesia. The mtDNA can tell us the contact happened, but not the details of how or why. Another unresolved question: why did only one group successfully leave Africa? The evidence suggests Homo sapiens evolved around 300,000 years ago, but the out-of-Africa migration didn't happen until 70,000 years ago. Did earlier groups try and fail? Were they blocked by climate, geography, or competition with other hominins? Genetic evidence hints at a near-extinction event around 70,000 years ago, possibly caused by the eruption of Mount Toba in Indonesia, one of the largest volcanic eruptions in human history. The human population may have crashed to as few as 10,000 individuals. The genetic bottleneck visible in mtDNA might reflect survival through a global catastrophe that eliminated most lineages. If this is true, everyone alive today descends from that small group of survivors. We almost didn't make it. The patterns visible in ancient mtDNA are repeating today, in real time. Modern migration driven by climate change, conflict, and economic pressure mirrors the ancient movements etched in our genes. Consider, the same coastal routes that carried humans out of Africa 70,000 years ago are now traveled by refugees fleeing war and drought. The Mediterranean crossing that brought early Europeans to the continent now sees thousands attempting the reverse journey from Africa to Europe seeking safety and opportunity. Climate models suggest that by 2050, up to 200 million people could be displaced by rising seas, desertification, and extreme weather. This would be the largest human migration since those ancient dispersals. And just like 70,000 years ago, these movements will reshape genetic diversity, languages, and cultures. There's a bitter irony here. The very populations most vulnerable to climate displacement, Pacific Islanders, coastal communities in Bangladesh, sub-Saharan Africa, are descendants of the most successful migrants in human history. Their ancestors crossed oceans, traversed deserts, and adapted to every ecosystem on Earth. Now, they're being forced to move again. Modern genetics also reveals ongoing mixing. A 2018 study analyzing DNA from 23andMe customers found that Americans are far more genetically mixed than they identify culturally. Racial categories, which have been used to divide and oppress, don't reflect genetic reality. There is no genetic basis for race as a biological category, only gradual variation shaped by geography and history. Yet the myth persists, sometimes bolstered by misreadings of genetic data. This is the danger of genetic determinism, the false belief that our genes determine our destiny, our intelligence, our worth. mtDNA tells us where we came from, not who we are. The most important parallel? Humans have always been migrants. The borders we've drawn, the categories we've created, the divisions we maintain, these are recent inventions. For 200,000 years, humans moved, adapted, and survived by cooperating across groups. The mtDNA map is a reminder we're all part of one continuous journey. So what can we take from this story? First, our shared origin is not ancient history, it's visible in every cell. Mitochondrial Eve isn't a metaphor, she's a genetic reality. Every person alive today carries a molecular record of humanity's journey from Africa to every corner of the planet. We are, quite literally, one family. Second, migration is not an aberration, it's the human condition. We evolved to move, to explore, to adapt to new environments. The impulse that drove our ancestors to cross oceans and mountains is the same impulse driving people today to seek better lives elsewhere. Understanding this doesn't solve modern policy challenges, but it should inform how we think about mobility, borders, and belonging. Third, genetics can answer where and when, but not why or who. Your DNA can trace your maternal lineage back thousands of years, but it can't tell you what those journeys meant to the people who lived them, or how you should identify yourself today. Science provides data, meaning is something we create. But here's the question we should be asking: if mtDNA could map the past, what is it telling us about the future? Every day, new mutations appear in human mtDNA. They're mostly neutral, neither helpful nor harmful, but they're accumulating. Thousands of years from now, geneticists will analyze our descendants' mtDNA and trace the migrations of the 21st century, the climate refugees, the economic migrants, the displaced populations. They'll see new branches forming on the human family tree. The question is, will those future humans look back and see us as the generation that learned from the past, that recognized our shared origin and built a world that honored human mobility and dignity? Or will they see us as the generation that knew the truth but chose division anyway? The mtDNA doesn't just show where we've been. It shows where we're going. And right now, that destination is still unwritten. If this video challenged how you think about human origins, or if you want to explore the science of genetics and human history further, subscribe and hit the notification bell. We'll be diving deeper into the evidence, the controversies, and the mysteries that still remain. Drop a comment with the most surprising thing you learned. I read every single one. We're all descendants of migrants. Never forget that.